Yes, as John said, I am a member of AstraZeneca, and today I'll give you a very brief introduction to myself, a brief introduction to the problem that we face doing drug discovery, and then talk you through some of the projects that my group in AstraZeneca have worked on over the past few years. Having seen some of the early talks this afternoon, I'm kind of wishing I'd gone into a bit more detail, but please feel free to ask me for more detail after the talk. So my name's Jonathan. I've been with the company for around 5 years and down here in Cambridge for about a year, and the quantitative biology group itself is a multidisciplinary group with bioinformaticians, mathematical modelers, image analysts, and preclinical stats. So, I don't know how familiar people are with the pharmaceutical industry, but we have a problem. The problem is it costs too much money to make drugs, and it takes us too long to do it. And in the industry, you recognize this. To get one successful candidate for launch, we need to have around 25 ideas at the very early stage. And as we go progressively down the pipeline, drugs will fail for a variety of reasons. The further along that pipeline they get, the more it costs to get that far. What's most worrying is even at phase 3, these are the very expensive trials involving hundreds of people, many countries around the world, the drugs still fail. Around 50% of them fail because they don't work. And this doesn't necessarily mean that they're completely ineffective. It just means that they're not as good as the standard of care, or they're not sufficiently better than the standard of care to justify prescribing them. They also fail for safety reasons, and some just because there isn't the right commercial profile there, but most of them fail because they're not good enough. To try and get around this, we have to make, as an industry and as a company, much better use of our information. And quantitative biology sits within the Discovery Sciences organisation at AstraZeneca, which is where AstraZeneca has centralised all of their early stage drug research support machinery. So everything from sample management, high throughput screens, reagents, assay development, dealing with externalization of our sciences, medchem expertise and structure and biophysics, and my own quantitative biology, with the idea being that a drug project can interact with us, the project can be handed off quite well. Eventually we'll kind of hand it off to the more preclinical and clinical research parts of the organisation. But and along the way, we're trying to make a much better use of all of that information, make sure that the assays are well purposed to answer the questions that we're interested in. So, now onto the actual projects. So this is an example taking some initial data or some initial model that was developed in vitro and actually making in vivo simulations and then progressing that on to simulating a treatment protocol for an oncology drug. So the DNA damage response pathway is quite an attractive target for oncology, as Mark suggested earlier, because many tumours have some kind of defect in this pathway. It's what allowed them to generate some kind of tumorous lesion in the first place. And broadly speaking, we can understand DNA damage response as being through two pathways. We have the ATM-driven pathway, which deals with repairing double-stranded breaks in the DNA, and the ATR pathway, which deals with repairing single-stranded breaks in the DNA. And the idea is these kind of molecules act to recruit all of the vast machinery that helps to repair the DNA in the case of damage. So we add an ATR inhibitor, we cross off this pathway, and you can see that this, we cross off this pathway, but there's still the functional repair pathway driven by ATM. However, many tumours have some kind of lesion, as I said, in that DNA damage response, and so if we can go into an ATM deficient tumour with no functional ATM response, if we inhibit ATR, the cell has no functional DNA damage repair, and as you can see from this data on the right, as we give successive doses of this ATR inhibitor, the damage, gamma H2X being a DNA damage marker, grows over time. The points are experimental data, lines of simulations, which will be the same throughout much of this talk. And as we give ascending doses of this drug, you can see that the tumour growth slows progressively until we can eventually more or less arrest tumour growth at the highest levels. To kind of get a better understanding of what's going on, and particularly to deal with more complex experimental data like washouts, we turn to a mechanistic model of the cell cycle, which you can see over on the left-hand side. My left, your right. My right, your left. So we start with healthy cells. And I should say these are healthy tumour cells. They're well functioning, they're replicating quickly, so they have G1 phase, they then go through S phase, G to M, and at this point they'll divide, go back to G1 phase. As they go into S phase, they can accumulate, or rather start to sense, the DNA damage. At this point, they will either be driven to apoptosis, or they'll repair themselves, and the ATR inhibitor will inhibit this repair process and hopefully keep them in the damaged state for long enough that even their deficient apoptosis sensing mechanisms can actually kick in, drive the cell down apoptosis, and remove it from the tumour. This model quite well describes kind of monophasic dosing, where you just put the compound on. Damage is accumulated. As you increase the dose, cells die quicker and quicker, the cells proliferate less. But the model can also quite well reproduce washout data. So this is where you put the compound on for maybe 24 hours, then you change the media in the well, wash the drug off. You can see that the damage signal falls off, and the model captures this trend as the cells start to repair themselves, and you can see we have a brief arresting of the growth, but then as the drugs wash out, exponential growth takes over. But again, as Mark alluded to, I'm really glad that you gave this introduction earlier. What we're quite often interested in doing is combining therapies together, and so the obvious thing to do when we have a drug which inhibits DNA damage repair is to induce a lot of DNA damage via ionizing radiation. We extend the model slightly with these states that represent DNA damage, and you can see that this manifests as an almost immediate increase in the number of cells that are gamma H2X, that DNA damage marker, positive, that then slowly repairs over time, and the model reasonably well tracks this in vitro. But what we can then look to do is couple it to a slightly more primitive version of, again, the model that Mark showed earlier, where we have this kind of, and this is a model of a xenograft tumour. So there's a proliferating shell around the outside where the tumour has sufficient nutrients to grow effectively, and then a necrotic core where the oxygen and the nutrients just can't get in there. And this model is parameterized on what that radius will be, etc. But by fitting this model, we can quite well reproduce the growth of the tumour in the absence of any treatment, and in various combinations of drugs and ionizing radiation doses. And what we really want to use this for is to provide a way of designing better clinical trials without having to run them, because they're very, very expensive and we want to get it right for the patients. So the treatment protocol for this study that we're investigating is quite simple. On every weekday, the subject will receive a dose of ionizing radiation, and then on 1 day a week, we can give them a dose of the ATR inhibitor. And at very, very low doses, the tumour grows out quite well. It's kind of kept at bay by the radiotherapy during the week, but over the weekend, it's growing out quite dramatically. But if we combine it with increasing doses of drug, we see that we bring the tumour under control. But the nice thing we're able to show through the modelling is if we give the treatment after the radiotherapy rather than in the middle of it, then we need a much lower dose, almost half the dose in fact, to control the tumour growth, which is better for the patient. It involves less side effects for them. It's also cheaper. We're using less drug to do it. And because we can investigate this computationally, we don't have to go through running very many complex clinical trials to try and figure out which exactly is the best way to dose this. So we're now changing disease areas, and one of the nice things about where I sit in the organisation is I get to see projects that sit all across the different disease areas that AstraZeneca does. So this is looking at safety for a CVMD, respiratory project looking at severe asthma. So CXCR2 is an important receptor in the response of neutrophils, and so the idea is we can perhaps treat asthma by inhibiting this receptor. As part of the safety studies, they did a study where they looked at neutrophil counts, and at day 4 we see a clear rise in IL-8, which is a ligand of the CXCR2 receptor, but also a clear rise in GCSF, which is related to neutrophil function, and this clear drop in the neutrophil counts at day 4. It's recovered by day 14 when the drug's washed out, so it's not a terrible thing, but it's still quite worrisome that we're seeing this neutrophil count drop. And the team didn't know what was happening, and perhaps more importantly, they didn't know what was happening in the bone marrow, which is very hard to get access to clinically because it involves taking bone marrow samples, it's unpleasant for the patient, and yeah, so what we set out to do was figure out, can a mechanistic model of neutrophil dynamics help to answer this question? So this is kind of a summary of what we knew about the neutrophil biology at the time. Neutrophils proliferate and mature in the bone marrow. GCSF promotes this. When they're mature, they start to express the CXCR2 receptor. This can bind to IL-8, and this will promote the migration out into the blood, where the neutrophils will circulate, and then they'll be cleared from the system by the natural degradation process, and our drug will hopefully interfere with this somewhat. When we translate that into a slightly more rigorous view, we end up with quite a complicated model. But the core of it is this neutrophil proliferation model showing the maturation through various stages, development of CXCR2 receptor release into the blood. GCSF kind of promotes the proliferation of these neutrophils. IL-8, when it combines to the CXCR2 receptor, promotes migration. And the drug, which we have driven by a PK model, but interacts with the CXC receptor, blocks IL-8, stops it from promoting migration. The resulting model, thanks to the quite large amount of data that we had, was actually broadly identifiable. I'm not going to claim every single parameter in there was identifiable, but we could relate most of them to individual pieces of data. So this was calibrated on some steady state knowledge of what was happening in the bone marrow from a number of studies. Also, a great many studies of GCSF injection, and the steady state values from the clinical trial that I showed you on the first sort of slide of this section. To validate the model, we used the full data from the trial, which you can see down here. So the points are data from the trial for the control arm in red and the drug arm in blue. The trial included an exercise challenge, which is this small bump here that we captured quite well, and a GCSF challenge, which is this large transient here, which we also capture. So a reasonably confident model is describing the existing data quite well. We can then look, does it actually, what's the implications for the study as we ramp the dose up and up and up. And it's worth pointing out that the clinically effective dose that we thought we'd be using is somewhere around here. The neutrophil effect saturates somewhere around 200 or 500 milligrams of dosing at around 40% of the control value, which quite nicely agreed with CXCR2 knockout in mice, just as an additional piece of validation, but that wasn't in any way used in this modeling. So this suggests that the compound is quite safe to dose unless the patient is already suffering from a low neutrophil count, but the target patient group actually has an excessive neutrophil count as a neutrophilic-driven asthma patient, so we're quite confident that will be okay for them. This next example is looking at one of those cases where a drug fails. In this case, where it failed in phase 3. This is Alzheimer's disease drug and a gamma secretase inhibitor. And this is semaglutide, which was a Lilly compound that failed, phase 3. We get some kind of indication from this clinical study where you see we give the inhibitor at time 0, we see an initial drop in A beta, which is a protein that's associated with the onset of Alzheimer's disease, but by 12 hours we've reached the baseline. By 24 hours we're still well above the baseline, and what's more, you can actually show that this drug, which we think should be inhibiting the production of A beta, is actually causing a net increase in the amount of A beta over 24 hours. If we look in vitro, we can see several different gamma secretase inhibitors at different doses, and they show this kind of intermediate rise in the amount of A beta being produced. The fact that we're seeing this across many different inhibitors with different chemistries suggests it's actually to do with the inhibitor itself, it's due to the target of the inhibitor itself. It's not some weird side chemistry of the drug. It's actually to do with what it's doing, and it's not clear why. The system is nominally quite simple. Amyloid precursor protein is cleaved by beta secretase, is cleaved by gamma secretase, we get A beta. When we look at the biology, we see it's a little bit more complicated than that. And this is kind of a snapshot of just some of that biology. So you've got the amyloid precursor protein at the top, beta secretase, gamma secretase, A beta. But there's also this alpha secretase cleavage, there's some other weird stuff over here. If we distill this down to the essential elements to show this rebound effect, you end up with this model here, where the amyloid precursor protein is cleaved by beta secretase to C99, and then by gamma secretase to A beta, or it's cleaved by alpha secretase to C83, and then to P3, which we believe has no kind of implications for Alzheimer's disease. And also there's this cleavage here because the alpha secretase cleavage domain lives within C99, so actually alpha secretase can cleave C99 to C83. The model works. We can quite well describe the data. And so these are two separate gamma secretase inhibitors. Points are experimental data, the lines are the best fit. And I just want you to focus on the top curves for the moment. You can see that we capture this initial rise in effect and then the kind of fall as the inhibitor actually starts to work. So why does this happen? The fact that we're using a mathematical model lets us interrogate this and try different hypotheses. But down here over in the low dose regime, we get quite equal cleavage, a small pool of C99, everything is fine. We add in some inhibitor. This will cause a build-up of C99 because we're stopping that from being processed. This extra C99 ends up being processed by the alpha secretase to C83. This pulls alpha secretase away from cleaving the amyloid precursor protein. This then leaves more amyloid precursor protein, APP, available for beta secretase. And so in this part of the regime, we see more buildup of C99 than we would expect from a simple linear proteolysis cascade. And what this means is that actually that increased C99 can overcome the inhibition of gamma secretase by more substrate availability. Of course, eventually these cleavages will saturate. We don't get any more of that additional buildup, and at that point the inhibitor starts to work. Now I said to ignore that lower curve. That lower curve corresponds to the situation in the Swedish mutation of APP. So this is a familial Alzheimer's disease mutation that's very, very rare because it causes very, very early onset Alzheimer's disease. And it causes this by massively upregulating the beta secretase cleavage of APP. The system already starts in this saturated state before we even add any drug, and so we get this nice, lovely, clean dose response. And it turns out that because we have such a high signal caused by this very, very efficient cleavage, that the Swedish mutation was used as the screening background for an awful lot of the Alzheimer's disease drugs. Yeah. So we can get some additional validation of this. If we take a cell line, we transfect in more C99, you see that the system goes from this biphasic behavior to the more monophasic behavior. We can also so to reproduce this PK data, we took the PK fit to semaglutide PK, we took a fit to semaglutide in vitro data. All we fit to get the two models to agree was a scaling factor of the production and a simple first order clearance of A beta, and we can quite well reproduce their clinical trial data, which is a very nice suggestion. So we think we found why we see this A beta rebound, and this obviously has some important implications for other therapies in the area, both of which fortunately seem to be free of at least this particular trap. Gamma secretase modulators aim to change the balance of A beta species being produced, and so shouldn't affect the overall throughput, whereas beta secretase inhibitors will act above this particular fork and branch shift behavior, and so again should avoid this as a clinical issue. Now, sort of going more to more spatial data, or at least how we're starting to, well, not starting, how we are generating that in-house. And this is really around marrying up high throughput screening, which is very much the kind of workhorse that we use to do initial drug discovery. But it's we can screen many millions of compounds, but it's very low on content. We might just get a yes, this compound is active, or no, this compound isn't active. Nothing more. High content imaging can get us a lot more information, as I'm sure I don't need to tell many people in this audience. And coupled to this, a lot of the drugs won't be active against our target. The typical response rate in a high throughput screen might be 2%. That's a good high throughput screen. It can be a lot less. Obviously, or perhaps not obviously, but it's not entirely computationally tractable to do a lot of the image analysis on millions of wells worth of data, especially if you know that most of those aren't actually going to be any good. So can we eliminate these? Yes. This was a workflow that we developed in-house to allow us to cherry pick the positive wells, image those using high content imaging as the kind of output of their HCS screen. So we start with hex cells transfected with the target and a GFP tag. We can do a whole well fluorescent intensity read of these wells. We can then identify the actives very, very quickly from this whole cell fluorescence. If they're showing any kind of fluorescence, we're interested in them. If they're showing no fluorescence, we can immediately bin them at this point. They can then go onto the microscope to be imaged at 20 times magnification. And by doing this, we can keep the imaging automation in much better use. We can keep the high throughput screening automation in much better use because it doesn't have to wait for that much slower imaging step. And out the end of it, we get a much more useful data set. We see some nice stuff where this might actually be an effect that we're interested in, where we're seeing the target kind of co-located with the cells. We can also start to identify weird artifacts where a compound is crystallized, a compound has gone into suspension, and all of these kinds of things that would otherwise have to be eliminated through an additional counter screen. And by doing a supervised machine learning approach, we can even start to classify these into phenotypes. What this lets us do is do a much faster and better decision making from that high throughput screening by eliminating a lot of the false positives right there at the start. And also, we can immediately classify the compounds into 3 distinct phenotypes that they induce, and we can start to look at those right off the bat without having to run some kind of additional and costly counter screen, which is very, very good. But can we do better? There's no real modeling yet of spatial data in early discovery that's applied in any kind of routine way, but we are starting to get the measures for that. We have a high content biology group, which we worked with for doing that study. Other parts of the business are starting to look at things like MALDI-TOF mass spec imaging to determine compound distributions in tissue. We've started to experiment with some live cell imaging technologies, things like that. But we're really not sure quite how to kind of work with this data because it's not data that we've worked with particularly in the past. So with that kind of open question to the audience, I'll finish here. And hopefully I've left some small amount of time for questions.
Thanks, John. That was great. You see the range of ways maths is being used. Questions?
So do you collaborate with academics?
Yes.
In what sense?
In a variety of senses. So as part of we have a very well developed internal postdoc program, which typically involves funding for a postdoc for I think it's 2 years that will be in partnership with an academic collaborator. We're also involved in a number of co-written grant proposals with academic groups, the specifics of which I can't recall off the top of my head, but we do have them. There's also, and I really should have put a slide in about this, the Open Innovation initiative that we have within Discovery Sciences, which is looking at making some of our compounds potentially, or some of our automation available to academic groups to run studies that they're interested in. And if you want to know more, please get in touch with me.
What are the intellectual property implications of working with academia? Because, you know, I'm told drug discovery is very competitive, and there are certain things which, you know, clearly are company secrets. So how does that work when you have, say, a postdoc?
So it's absolutely a requirement for our postdoc projects that they will be able to publish. The project must not be, we will shut you in a box and work on a top secret research project for 2 years. You won't be able to tell anyone when you leave. And in fact, as a scientist working for AstraZeneca, I have a requirement in my personal objectives that's handed down from the head of early stage drug research that I will get my name on one paper a year. We're really trying, at AstraZeneca, we're really trying to push publication and academic openness everywhere that we can, because while there are some things that obviously we want to keep to ourselves for intellectual property reasons, a lot of the things that we work on are not that sensitive. And there's a growing recognition within certainly AstraZeneca, and I think within the industry as a whole, that we need your help, and to get that, we have to open up.
Starting to move into the discussion section, so we'll move on to that very quickly. I've got one more very precise one, finish with that. So, for example, your cell cycle model, is that in-house, or do you take one from the literature for that?
I think that was just in-house. It's literally, that was the model. There are no kind of equations for the different chemical signaling markers that lie underneath that, because we didn't have any data that would infer how those would differ. So it's, yeah, those parameters, the K1 to 6 on the diagram, are the parameters in the model. They're not masking a lower level model.
Thank you. So we are about to move on to discussion. Before we do that, can we thank Jonathan and all the speakers? I've actually enjoyed that.